About ready to start here? All right. Well, hi everyone, and thank you so much for joining us at our inaugural talk for UBCO's Diversity in Biology speaker series. We're super excited to have everyone attend wherever you're calling in from. This series is the result of a lot of hard work, and it would not have been possible without our faculty coordinators, Dr. Karen Hodges and Dr. Robin Young, and our graduate student coordinators, Shirley Sujie Yang, who's doing her master's in Dr. Clegris's Brain Cellular and Molecular Pharmacology Lab, and myself, Kat Fovell, doing my PhD in Dr. Ford's Wildlife Restoration and Ecology Lab. Shirley, Dr. Hodges, and Dr. Young are calling in from UBC's Kelowna campus, which is on the territory of the Syilx Okanagan peoples. And I'm joining in from Edmonton in Treaty 6 territory, which is home to many Indigenous peoples, including the Cree, the Tsuut'ina, the Blackfoot, the Métis, the Nakota Sioux, the Iroquois, the Dene Salune, the Anishinaabe, Saulteaux, and the Inuit. We acknowledge the many First Nations, Métis, and Inuit who have lived and cared for these lands for generations and continue to do so today. We invite you to acknowledge the Indigenous people who care for the lands you're on today as well. With us, we're pleased to welcome Dr. Jared Ali, who's currently an assistant professor in the Department of Entomology at Pennsylvania State University. He received both his bachelor's and his master's degree from the University of Delaware before moving to the University of Florida for his PhD. He did postdoctoral work at Cornell and also spent some time at Michigan State as an assistant prof before moving to his current position in 2016. I'll hand it off to Dr. Ali in just a second, but a quick housekeeping note. The audience is free to post questions in the Q&A throughout this talk. The questions won't be visible publicly until Dr. Ali has finished his presentation, but please feel free to post them as they come to you so that you don't forget about them. The seminar is being presented through Zoom's webinar mode, so you will not be able to turn on your cameras or microphones, but you can post in the Q&A. Because of the nature of the seminar series, you're free to ask Dr. Ali any respectful questions about his research or career path so far. So without any further ado, please take it away, Dr. Ali.
Great. Thanks for having me out here. I'm excited to give a chat about all the different things that we're doing in my lab. Everything in my lab perfectly seems to have a tacky alliteration from these bad bugs, pungent parasites, and toxic travelers. I'm going to talk a little bit about the context of cover crop cascades and crosstalks, but I think I'm overdoing it with all of the alliteration. But before I get into all my research talks, and especially seeing as it's pertinent based off of the topic and the nature of this series that I've been invited to speak on, I want to get and touch base with some of the events that have been influencing all of us. We've been living in this time of pandemic. Obviously, we're Zooming. There's some drawbacks. There's some benefits to that. For my home life, and many of us, we were trapped at home with families and loved ones. I was literally often getting peed on while I was answering emails and Zoom calls, while my other son was supposed to be in kindergarten running around the house. It was insane. And all of this to try to look professional and to keep things rolling. On top of this, just being trapped at home in the pandemic, there was even more upsetting things that were occurring. You know, just in the time of pandemic, you'd think that there might be more unity, but we were also seeing increased conflict and higher presence of social inequity and justice issues. And during this time, I did a lot of reading, and among some of the time that I had when I wasn't changing diapers or pretending to be a kindergarten teacher, I found some time to read some books. Among them, these are two I really appreciated and liked. And this is the New Jim Crow Law and the New Jim Crow and Sapiens. These kind of represented an interesting continuum for me. At one end, you have in New Jim Crow, this idea of these evolved constructs and barriers and issues that we have in North America, namely in the United States, that seem to be restricting others to resource and prohibiting equity. That's been happening over time in the United States. At the other end of this perspective, we have this global macroevolutionary phenomenon of our species. And you can see the kind of issues and constructs and barriers that we have just with our lizard brain that have allowed certain kind of inequities to persist and imbalances in our sense of diversity. So reading these books, these are two I can highly recommend, and a bunch of other ones I read, I kind of came up with some ideas and some questions. And along with these kind of questions, I developed kind of these aphorisms or take-home messages that I got from them. So the first is what is rare, what's diversity, and what's exceptional. And then kind of at each topic, I kind of go over 3 ideas and questions I had. What's it mean for your experience? What are the consequences? Is it good and bad? How do we put this all together? So let's start off with rare and rarity in general. So what is rare? Something that's really interesting about this idea of rare that people often don't connect are resources. And when I look at this symbol here, which I hope you all recognize as a symbol for a restroom, correct? It's funny that we recognize it, but over time, despite the fact that this is one of the most essential resources that all humans need, that literally we have to do every day and deal with, over time, across cultures, across countries and barriers, there's been prohibitive and exclusionary laws for who can use this, whether your sex, by the color of your skin, the disability you may have, the gender that you associate, that we've been providing a barrier to this resource of restroom. And rare individuals in various sectors, as they've been trying to increase or be on the frontier, have always had to deal with this lack of a resource. There's a lot of other things that come up with bathrooms and rarity that I've been thinking about as well. When we think about this kind of idea that I often even struggle with, similar to this parent here, as a male who often tries to help and provide with my family, there's been a huge prohibitive issue. When I go to the bathroom to change a diaper, there is no resource there for me to help out with my children. Often it's really interesting if you take this a step further to think, if I'm out to eat with my wife or my partner, the assumption is that regardless of the nature of the meeting, whether it was a business meeting that she hosted and planned, the assumption is that she should be the one going to the bathroom. The assumption is that my place is at the table and her role is to be there. So when we think about resources, it's funny to realize that some of the issues with them assume certain people have to occupy a specific position or role just because that resources isn't there. On another level, resources like bathrooms have also been a place for people in the frontier that are rare, where they get relegated to this place to provide for their families. If you're the lactating member of the family, it's been often this place that you're shoved off into to use because the resources aren't there to be a lactating family member, suggesting that at different systems and different industries, different academic institutions, it's presumed that those members of the family aren't supposed to be taking care of their family or providing natural functions, food for their family. On top of this, when you put these ideas of resources, availability, and you think about a member that's on the pioneer early on, in this picture, in 1927, there's only one female here, Marie Curie. And she's among all of these famous scientists. You have Albert Einstein there, all of the biggest names in physics. At this conference, you can imagine that it's highly probable there wasn't even a restroom for her to use in the building behind her. I don't know if any of you have noticed this, but if you've ever gone to an academic institution, you've come across a building where there's bathrooms, particularly older ones, where there's restrooms every other level, where it goes male, female. The reason that is so is because in prior years or in the past, they were all male restrooms because it was assumed women didn't have a place there in these academic institutions. And I've noticed this at almost every institution I've been to, if I've gone to one of the older buildings. So if you just imagine what Marie Curie was going through, whether there was even a bathroom for her after her talk, the questions and things her colleagues could have been dealing with while she had a full bladder thinking about, where am I supposed to go? These are the kind of challenges that you have as a rare individual that others might not appreciate, what it takes for you to be at the table and the type of barriers you're dealing with day to day just to come and be somewhat on equal level or playing field with everyone else. So 2 takeaways from this rarity is that often, you're going to hear things that are naive and often insulting questions. On the other end of this, it means resources that are available to others may not be there for you. And it also means that you're meant to occupy a certain role for better or worse. So a part of this segues into the next topic, which is diversity. So as we try to increase these or make certain individuals less rare, we have this idea of increasing diversity. So what is diversity? As biologists, we've all been trained in all these diversity of matrices and indices we use. We know that diversity can vary across the genetic level, the species level, and at a larger ecosystem or community level. But more and more across all of the sectors, from industry to academia to at the institutional level, we've been trying to make these pictures of diversity, where you simply have this color spectrum of all the colors of the room, all these diverse skin tones. And if we can achieve this, we've truly achieved diversity. A problem with this is that although it superficially gives you a sense that you've established a diverse room, what's the real value of this? On top of it, outside of just having a diversity of skin tones in a room, we often miss other perspectives and other metrics of diversity that you can't easily observe by just looking at someone's skin tone or looking at them, right? What's even more harmful in this idea of just adding the numbers and getting to certain benchmarks is that we know as biologists, we've been trained that diversity is the key to everything we see on the planet. And all the taxons, all the taxonomic diversity we see, all the species, everything here is because of this inherent diversity that's key to life itself. Yet we still typically don't think about diversity beyond just adding a brown person to the room, inviting a brown person out for a talk, or increasing numbers of that underrepresented human. It's interesting because we know that there's power and function in diversity, and there's even terms, functional diversity. And that means you have to really understand how you utilize inclusion and matriculate these diverse individuals into it, and really utilize diversity as a strength. Really understand the mechanisms that allow diversity for either radiation or success of a department or a program or improved outcomes. And that's something that's often missing with these often shallow metrics we take for inclusion. Now, a part of moving forward with diversity, increasing rare individuals and resources, comes along with this idea of exceptionalism. And what is exceptional? At the time in the early 2000s when I was getting my PhD, there were technically more Black males in prison in the state of Illinois alone than Black males enrolled in undergraduate degree programs across the country at state university level. Even more so, I did more number crunching from data from 2018, 2017 that shows I would have, if I was just growing up in almost any state in the country, I have more than twice as likely chances to be incarcerated and in prison than I would be to be in a graduate program. So by all measures, I am rare. I'm an exception to the rule. A problem with this idea of people being an exception to a rule or being exceptional in general is that it leads to all of these examples where people from different backgrounds or diverse, challenging histories or personal history. It means that there's this concept that anyone who looks like them or falls under that category of marginalized or minoritized person who succeeds, anyone else who looks like that or is that way didn't succeed because they aren't trying hard enough, right? And it means, oh, if Jared could do it, all the other Black people should be doing it. It just means they're not trying hard enough. Well, I know firsthand that this isn't necessarily true. My brother, similar background, same, we came from single mother households, first generation. While I was getting my PhD, who's struggling to go into college the same way I did, he would call me on the phone like, how'd you deal with this? We don't have enough money for textbooks. How did you do that? I was like, well, you go to the library, you check out first editions. They're old, but you can usually pass the class. What do I do when classes end and they break out and I don't have a place to sleep because the dorms are closed? Well, I usually found a friend's couch to crash at, or I slept in the back of my car, literally. I missed, things like missing a bus. I used to walk 8 miles when I missed the bus to get to my night calculus class. And so I told him to do all that. That's what you got to do. That's just the fact of life if you want to make it. This isn't fair for everyone to think about these lack of resources. And I know this is not true, and there's issues with this concept because, and I'm going to say a trigger warning for anyone that might be affected by this, but my brother, his freshman year of college, took his own life. And so it meant that there's a big issue with this idea of exceptional, or you have to be exceptional to make it. It's because it leads to resilience rather than resources. I was resilient. I wasn't provided resources, and that's the only way I had the only venue for me to make it. My brother couldn't handle all of those pressures, and understandably, it's a lot for people. On top of that, it leads to this idea that others can think that those that are trapped at the bottom are choosing their own fate, disregarding the lack of resources, the lack of understanding to matriculate diverse groups together and utilize them, as well as this idea of just they have to work harder. So how did all these kind of ideas come together on diversity to influence my sense of insects or relate to insects overall, since that's the study and field I'm in? Insects technically end up being this perfect model for diversity. And for some reason, humans just can't seem to get it right. Right now, for every one of you, there's 200 million insects out there. On top of it, when we look over the history of humankind, there's multiple species of humans since we've been on this planet. Now there's only one. For insects, on the other hand, you see that they've radiated more and more, and there's estimated to be more than 1 million species of insects out there. Something about Homo sapiens seems wrong to me and off in our tendencies and our relationship with diversity. And you can see across fossil records or any history that you have of wherever Homo sapiens, our species, occurs, we reduce diversity at almost every taxonomic level in every group. We cut down trees, we kill off species, we often killed off any other human species that was present. It seems as though and once we get rid of all of that level of diversity, we start to fight among ourselves, right? And then on top of it, if you look at insects, on the other hand, wherever they go, they often are creating new niches and forcing diversity where they go. Something about humans is off. And it seems that whether it's Hutu or Tutsi, Israel or Palestine, hanging up flags of separatism and using that as a way to divide countries, or literally building walls along imaginary borders to separate peoples. Humans don't seem to understand that diversity is a measure of success. Insects seem to be pretty good at it. And there might be something in there that we need to learn about this functional diversity, how to successfully go on and how to be sustained. There's a lot that insects do that we have wrong. And I've been learning a lot from them. But how did I get into insects in general? Well, that's an interesting story in itself. It kind of starts off with my adolescent background. Because of some of the hardships of my childhood that I described earlier, I chose to go to this Quaker boarding school. So we moved a lot, and this picture gets older and older every time I show it. It already looks like it's centuries old, but this is back when we used real cameras and, and these photographs would fade. And so here's me actually there. And you can also see how this is so old because under that hat, I had tons more hair than I have today. But what's crazy is I picked a boarding school because my family moved a lot. We were getting evicted, and I was able to pick schools to get scholarships from. And I found out that there are these things called Quaker boarding schools where I could live. I wouldn't have to move around when my mother moved around. And so I found one that was awesome. I also love the fact that you called your teachers by your first name, super liberal, open environment. And it was amazing for me. But I still wasn't getting everything I wanted out of it. And apparently, instead of staying in school, in high school, I decided to literally drop out. I sold a bunch of my records and all of my belongings to actually a bunch of the teachers that were on dorm. They had no idea what I was doing, selling all my David Bowie and Miles Davis records. And I got my money together and I left Pennsylvania, hitchhiked and took buses across Canada. And then I had this plan to go down to Mexico to hang out. Luckily, I had an interesting stop in San Diego, where I met Edward Brinton, or Ned, who has passed away a few years ago. And I met Ned because when I was in Canada, actually, it's funny, I'm talking to British Columbia. My friend Gwen was living in Nelson, BC. She had graduated a year before me. And she said, Jared, we got to stop off in San Diego. My grandfather's there. He's a biologist. And she knew I was really into science. And she's like, you should check him out. And we should stop there, get his four-wheel drive before we go into Mexico and kind of hang out in La Jolla before we head on the journey. So I stopped off there, and every morning I had really interesting conversations with Ned. We had he would make these huge bowls of cereal, a huge cup of coffee, and he and I would talk all morning. And I found out that he worked at Scripps Institution of Oceanography. And I was like, I don't understand it. It seems like you've traveled the world. You have all these cool pictures, research, you live in this beautiful city. Like, how's it possible you do that being a scientist? And he said, Jared, you should come down to the lab. He brought me into his lab and he showed me these tiny little invertebrates, which are krill. And he convinced me easily that these things are connected to all these different trophic levels. And a part of his research studies were understanding and modeling krill populations to not only track and monitor whale migration, but also he developed models that were associated with climate change patterns. So he was taking these tiny little organisms that most people weren't seeing, traveling the world, making these amazing stories and connections. And he was able to do this with the power of a PhD. So I realized maybe I should go and go from a high school dropout to my PhD. And I got what we called my good enough, damn it, which stands for good GED for me. And I went GED to PhD. I ended up going down to Mexico, long story, crazy stuff. I ended up back in Delaware, just outside of Pennsylvania. And I wrote letters to the University of Delaware, and they said if I take classes at a community college, I told them all about my passion for biology, this crazy journey I've been on. They would matriculate me as a biology major. I stayed on at University of Delaware, where I literally ended up becoming the face of entomology during my time there, and studying these tiny little invertebrates, similar to what Ned was doing. I ended up going back to Delaware more recently, about a few years ago, where I got the Early Career Award for the College of Ag from the university. And here's my advisor, Douglas Tallamy, who was an amazing mentor to me while I was grimacing accepting this award. I grimace a lot when I'm happy for some reason. So anyway, I went on from University of Delaware. I had a bunch of other really great advisors, from Doug Tallamy to University of Florida during my PhD with Lucas Stolinsky, also first generation immigrant from Poland, and first generation college graduate, PhD, and so on. Then I went on to Cornell University, where I did my postdoc with Cornel Anurag, also first generation immigrant from India, and not first generation graduate though. Then I went on to Michigan State, where I accepted my first faculty position. But because of all of those things that were going on with my family as well, and my brother who had just committed suicide and lost some other family members, I took an opportunity to go back to Pennsylvania, where actually my brother was his first year of college, was at Penn State University. So it made a lot of sense for me to be able to go back there. On top of it, they had this great chemical ecology program there, which I'd always dreamed of joining. On top of learning how to fish from my PhD advisor and taking care of chickens like my postdoc advisor, I also learned a bunch about plant herbivore interactions, which kind of changed my interest in these multi-trophic stories I'm going to talk more about. So both these advisors kind of convinced me that there's these cool array of defenses that plants have to cope with all of the organisms that want to take advantage of them. And since they have this diverse array of things munching and chewing and attacking them, plants evolve these ways to not always have their defenses on, but induce them when they're in stress, when they're struggling. That way they don't waste resources, they can allocate them to growth and then turn them on when something's bothering them. Now, in my field of study, there's a bunch of different ways to study this. 3 of the ways and topics I'm going to go into today revolve around defense signaling, namely how plants can release volatiles to influence multiple trophic levels, trade-offs or crosstalks in phytohormone defenses. And then we're going to talk about how also insects can not only deal with these plant defenses, but can circumvent them and steal them for their own benefit. And so we'll go through these 3 little stories that we have in the lab, and I'll give you a little background about each one before we get deeper into each topic. So the first one's these defensive signals, particularly volatiles. We all can often appreciate the smell of grass when it's freshly cut, right? You can go outside and you immediately smell this. That's because there's this release of metabolites that were in that leaf tissue that's now out in the air circulating, and your nose can recognize that change. In an analogous manner, when a plant gets chomped on by an herbivore, there's this change in the volatile profile, and subsequently, higher trophic levels have started to eavesdrop on this and use this as like a plant's cry for help to find a reliable source of food in this herbivore. Now, there's a bunch of assumptions and context around this. First, we usually talk about this as being good for the plant and for the predator that responds to it. It's usually described above ground rather than below ground. And it usually just focuses on the plant, the herbivore, and the natural enemy, excluding all the surrounding organisms that could be eavesdropping on this information. Part of work I did early on during my PhD was evaluating these kind of indirect herbivore plant-induced volatiles below ground and how herbivores could influence natural enemies, which are these things that we study in the soil called entomopathogenic nematodes. Now, instead of predators, EPN or entomopathogenic nematodes have been used for biocontrol for soil organisms for quite some time now. They're these tiny worms that have this infective juvenile stage they're looking around the soil for an organism. Once they get in the soil, they puke up a bunch of bacteria. The bacteria and the nematodes grow together, eating all the resources of this decaying, rotting insect. Once the insect's resources are depleted or all the kegs are kicked, you have this new party of new generation of infective juveniles running off into the soil looking for their next party. Now, this has been seen as a great biological control tactic, but there was some desire to improve this by using herbivore-induced plant volatiles below ground. So part of my PhD, we had these root zone olfactometers where we could put larvae onto the roots of plants. Here, I worked a lot in citrus with a citrus root weevil. And then we could apply nematodes to the central arm and see to which arm they would be attracted, whether the plants had root herbivores on them or not. And the general questions were, did we know if herbivore-induced plant volatiles were playing a role in protecting plant host? Well, through a series of work I won't go into because I think it's been too long since I can't keep talking about my PhD forever. We found out that yes, the volatiles could influence these nematodes, but they also affected additional trophic levels. What I had found is that doing a series of experiments, not only were EPN affected, these beneficial nematodes, nematophagous fungi could be responding to this. We also found that competing nematode species that also feed on bacteria would be responding to this signal. We even found that plant parasitic nematodes would respond to these signals. So limiting it to just the third trophic level really changed our perspective. And when we zoomed out a bit, we realized this was way more complex. It also made me think a lot about information in general, not only that it can affect more than just who you're focusing on, but also that it might move bidirectionally. And this is because there's been more and more work showing that we're in this age of information, particularly with plants and herbivores. And we know that plants can respond to cues from insects, from their frass, from their saliva, that insects are responding to plant cues. We also realize that predators are responding to multiple signals in this context. We know that volatiles from plants that are stressed can communicate to other plants, so plants are responding to this. And what we find out overall is when we look at all this complex volatile signaling, it's all just a type of information. And when information gets released by one organism, it's just out there. And anything could respond to it, whether it's a good player or a bad player. And it really started changing the way I thought about utilizing and leveraging these signals. One thing that had come up in prior research is that plants could eavesdrop on odors from their insect damaged neighbors. This means when a plant gets fed on by an herbivore, it releases chemical cues to nearby neighbors that could then prime their defenses, getting ready for the attack of these herbivores that could be hopping to their leaves next. This has been found in lima bean, maize, tobacco, and sagebrush, and has been more and more common as it's been looked for. Something really lucky for me is I came across the work of Dr. Angel Helms, who became a postdoc in my lab. What was cool about what Angel was working on is that she was one of the first to find, or actually the first to find, that plants could eavesdrop on odors of insects. So plants could smell insect herbivores. This opened up a whole new idea for me that plants could be sensing and responding to all types of cues. Luckily, I was at a meeting in Sweden, and I stopped Angel and I started talking to her. I said, "Hey, Angel, I work on these nematodes. They kill beetles pretty well. And when they kill insects, they have this really strong odor. You think we could come up with a way to study this more?" Angel was happy, surprisingly, and wanted to join my lab. And we went further and got some USDA funding to go down this path. And what we knew is that plants can release odors towards a third trophic level in one direction. Our question was, could nematodes, these beneficial things, be releasing signals that could affect the plants? We took some cadavers that were infected by these nematodes and sampled their odors. And what we saw is that these EPN infested cadavers are releasing way more cues than our freeze-killed cadavers. We then developed this system where we could push air over entomopathogenic nematodes in the soil, push this onto plant roots, and see whether herbivores would grow bigger or smaller in response to these nematode cues. We were really surprised to see that these beetle larvae, these Colorado potato beetle larvae, were performing much worse in terms of their larval mass when they were feeding on plants that were receiving cues from these entomopathogenic nematode cadavers, more so than control cadavers or just plants with nothing in the soil alone. We also wanted to see if these were actually priming any of the defenses of the plants. And I'll talk more about phytohormone defenses now, but for now, I guess you could just understand, we looked for hormones in the plants to see if the plants were actually upregulating their stress responses. So we looked at jasmonic and salicylic acid here, and we found is that salicylic acid was being induced whenever we had these EPN cues around the plants. We also did some more work and found that this was primed over time, and that we did some sequencing, we did some work with the real-time PCR for markers for the salicylic acid pathway and saw that the plant was genetic expression was changing as well. We also wanted to see whether these cues could influence the behavior of the herbivore. So we took these cadavers and we had an arena where we could have a plant that had either these nematode-killed worms or the ones without the nematodes, and we would see whether a gravid female that had eggs would choose to lay her eggs differently on these plants. What we found is that EPN cues reduced oviposition of these Colorado potato beetles, where there were way fewer eggs laid on plants that had this EPN cadaver cues compared to any of the other plants in our arena. So our take home from this is that signals and below ground signals can be bidirectional. They can move up and affect higher trophic levels, but also plants can actually eavesdrop on their predators and change the way that they're responding. The crazy part is that it can influence plant response, can alter herbivore behavior and performance. And we're looking deeper into this, and we have a few more papers where we're trying to understand how this works with below ground herbivores as well. So I brought up in the study some interesting trade-off hormones. I talked about salicylic and jasmonic acid. These are these plant hormones. These play an important role in these plant defensive pathways. And analogous to maybe testosterone or estrogen, things that can have this attenuating response on one another, plants have salicylic acid and jasmonic acid. And what's interesting is the upregulation of one causes the attenuation or downregulation of this other pathway. And when we think about this for plant herbivore responses, some herbivores actually induce one pathway, while other herbivores that chew on plants can induce the other. So we can see here where phloem feeding insects that have piercing sucking mouth parts induce salicylic acid, and chewing herbivores can induce jasmonic acid. When they co-occur on the plant, you have this asymmetrical effect on response, where some herbivores do worse and the other herbivore can do better. Now, there's a bunch of assumptions and context around this as well. We see that there's this crosstalk, and what we find is we think crosstalk interactions are abundant. However, it's rarely evaluated in a way that you could manipulate the pest or insect behavior. Furthermore, we rarely find ways to leverage these crosstalk events and these phytohormones in agriculture other than just dumping hormones on plants and seeing what it does. So I began looking for ways that you could change context of plants to modulate and affect their hormonal cascades. And we've done this through using cover crops. And I have a bunch of work behind this with a bunch of students and postdocs. I've been working on these projects funded by a few different resources. And what we're trying to do is take cover crops, which come in prior to your cash crop, use them to change the soil fauna. Namely, initially we were focusing on arbuscular mycorrhizal fungi, see how each cover crop could affect this legacy, which then change the way a plant could respond either hormonally or through volatile signals, and then look at a bunch of different herbivores and pathogens to see how each cover crop could change this crosstalk to either benefit or harm the plant. Now, one reason this is important is because we know plants have a bunch of different things they're coping with and dealing with. And because of that, sorry, I'm moving some Zoom stuff around. And because of that, these plants they're getting pulled in different directions chronically, whether it's a gall maker or chewer or something that's phloem feeding or mining through their leaves. Plants have to get pulled in all these directions. What's interesting though, is there is context in the soil that we can often change because there's so many key players there, some beneficial, some harmful, but we were looking for ways that we could modulate them using beneficial soil organisms. And since plants get pulled in so many directions, towards one pathway or the other throughout their life, I've started to observe throughout this that there's some things that we could take advantage of. Early work during my postdoc, I found that when you had two occurring herbivores on a plant, like a monarch butterfly larvae and these oleander aphids, monarchs would do way better when aphids were present, but only on a particular plant species. We also did a bunch of work with feeding guild interactions where we had aphids and beetles on plants, and we found that the behavior of the organisms would change based off of who was on the plant first. More recently, we've been looking at cues from insects and seeing that cues released by one insect, such as the fall armyworm larvae, would be great for that larvae to feed on the plant, but if we isolated these chitinases and put them on the plant, it would be harmful for these other herbivores. So we started to ask, how could we actually take advantage of these trade-offs? And which ones weren't we recognizing? And which ones could we control? At the soil, there's one major player that we started to focus on, which was arbuscular mycorrhizal fungi. And this is great because they can influence these pathways such as salicylic acid, jasmonic acid, and also communication signals depending on their early phase of infection or being inoculated on plant roots. AMF are cool also because they can alter herbivores in multiple ways. They can change the herbivore's response to the plant by just affecting plant vigor overall, or they could be changing things like the plant defenses. The hard challenge in getting beneficial organisms in the soil is that the way we do agriculture now, we're often disturbing this, whether we're dumping chemicals, growing plants in monocultures, or tilling the soil up to really change and affect all of this. It seems that it's challenging to establish these beneficial organisms. So cover cropping became a really interesting way to manipulate these organisms. What we wanted to ask is, if we measured maize response to various pests and pathogens after different cover crops, could we see how these legacies either improved pest resistance, if each pest would respond differently to these legacies, and what effects could these legacies have on pathogens? To understand cover crop in general, you usually plant your cash crop in the summer, and then once you harvest that, you'll put a cover crop in the fall. This often builds soil health, retains nutrients, prevents erosion, and can suppress weeds. And then before you go back and plant your cash crop again. Although we knew of all these kind of abiotic benefits that cover crops could have, we're only recently realizing that maybe we could select cover crops that could be non-mycorrhizal, affecting that soil community, or mycorrhizal, and then see this cascading consequence on the plant metabolites and subsequent pests and pathogens. So we did this by collecting soil from this huge plot we have. We have this cover crop cocktail site, a lot of acres out here, where we have each successive cover crop growing, followed by our maize cash crop. And we could go out there and collect soil to do some in-house in the greenhouse experiments. We did a bunch of field work as well, but I don't have time to go over that. And it gets super messy. This is a lot of work by a bunch of different students. But I will go over some of the soil legacies effect that we did in the greenhouse. So we went out there, collected soil. We were then able to take the soil back into the greenhouse, pot them up and put maize in them, apply pathogens or herbivores, have them all contained on the plants for a while, look at volatiles that could be released in the shoots of the plants, so the odors of the above ground portions. And we also developed ways to sample odors of the below ground portion of the plants as well from the roots. And on top of that, and looking at the herbivore performances or pathogen performance, we also measured a bunch of different genes, things associated with the JA and SA pathway like LOX and PR5. Also look for things like these maize protease inhibitors that can really change the amount of nutrients that herbivores can get from a plant, as well as some things associated with these benzoxinoids defenses that grasses have, this BX1 gene and this indole associated gene that also affects the benzoxinoids pathway. On top of that, we also looked at these terpene synthase genes that are responsible for some of these volatile profiles that plants produce over time. When we first started this off, we were happy to see that the arbuscular mycorrhizal collination differed depending on cover crop. And here you can see our stained roots example. And we were seeing that, yes, cover crop was imparting differences on our AMF community. Now, for simplicity of this, I'll go over all of the performance effects of herbivores and pathogens across the cover crops. But for the genetic work, we ended up focusing namely on radish and triticale because they had this huge contrasting difference. And across all of our studies, the biggest differences was usually through radish and triticale. So I'll focus on those for a lot of these expression or volatile changes that we noted. When we first did our herbivore performance with aphids, surprisingly or not so surprisingly, there was no effect on aphid growth. And I was kind of like, well, that's great. Nothing happened there. But what we did see across cover crops is depending on whether they were feeding on radish or triticale, their terpene synthase genes were changing the way they would express. So we were seeing a really different or induction of this terpene synthase genes when damaged or suppression when they were damaged compared to undamaged, but only when they were feeding in radish soil, legacy soil. When we moved below ground to look at corn rootworm responses, what we saw is that, yes, there were differences in corn rootworm performance by cover crop, where it seemed as though these root larvae were doing much better, surviving much higher on pea or radish soils compared to triticale or fallow. When we went forward to look at some gene expression, we saw that there was some effects of the maize protease inhibitor, where it seemed that undamaged plants grown after triticale soil seemed to be expressing a high rate of this defensive gene, meaning that they were initially having to deal with this. And this is interesting because the triticale soil is where these herbivores were doing pretty poorly. In addition to this, working on below ground organisms is tricky, and finding them is even trickier. So we've been developing ways to identify these things occurring in the soil and in the wild, and we're using these volatiles to predict their presence. We have a bunch of different arrays where we're trying to increase the amount of replication to feed this machine learning algorithm, so we can develop sensors or probes where we can detect and tell which herbivore is there. Right now, we've gotten to this point where we're using all these classifiers, and we've been able to predict the occurrence of larvae with 80 to 70% accuracy. And we're seeing a bunch of different terpenoids are playing important roles in this. We're still ramping up this, and this is all part of work that's in progress from Olivia Trace, a PhD student in my lab. We next moved on to fall armyworm, and what we were surprised to see is that for fall armyworm as well, as opposed to the aphids, we saw that there was another effect on their weight gain and how much tissue they could feed on when they were feeding on plants that came from any of these soil legacies. We went further to look at this, and we saw that the soil type altered the induced defense of this MPI gene, but not the LOX. We saw that the LOX pathway, this is something that's also associated with the jasmonic acid metabolism. We saw that although there's induction, there was no difference between the radish or triticale soil in the plant's ability to respond using the JA pathway. But for that maize protease inhibitor, we could see that in radish soil, there was a clear distinction where it was getting induced, but on triticale soil, we weren't seeing that. We went further and looked at larval behavior to see if these herbivores could actually recognize the differences in these plants, particularly since on some of them, they were doing really poorly on. And we developed an assay where we could let this larvae released into the central chamber, see which plant it would be attracted to. And we found is that larvae avoided odors emitted from that maize plants grown after triticale. That's that plant that they did worst on. So there's some evidence for this preference performance interaction going on. So we saw that they were choosing triticale equal to a blank arm, and they would definitely prefer fallow, pea, or radish soils over that triticale plant. We sampled volatiles from the plants to see if these are actually what signals the larvae were utilizing. We detected a number of different signals, but we did see some of the main players where this alpha terpineol was much lower in triticale and higher in pea and radish. And then we also found this trimethyl tetratriene was way lower in triticale again and higher in pea, radish, and fallow. So we kept seeing that this consequence meant that there was something in the volatile profile larvae could use to make decisions about the appropriateness of a host or how well they would perform on it. So we see that these cover crop soil legacies were having dramatic effects on different herbivores in different ways. How would they then affect pathogens? We went forward and used Fusarium verticilloides to look at this pathogen. This causes a bunch of different issues from stem rot, seedling blight, and ear rot in maize. And it also produces these nasty mycotoxins that are bad for humans and animals alike. What we first did is took these soils themselves and saw how soil infused with this verticilloid Fusarium would actually affect maize germination rate. And what we saw is that germination rate was affected when Fusarium was present. So when we looked at the soils without Fusarium and we counted germination over time, we saw that they pretty much didn't resolve any differences. So without Fusarium in the soil, we saw pretty much equal germination rates. However, when we added Fusarium, again, we saw this crazy kind of difference, especially between radish and triticale, since we could actually manipulate radish and triticale compared to fallow, that triticale was now one of the worst performing in the presence of Fusarium, while radish was performing much better when Fusarium was in the soil. We looked closer at this to look at ear rot assays, and we could inject Fusarium by syringe into the cobs of corn. And what we saw is that the area of infection was much larger on that triticale soil than when these plants were grown in radish soil. So it's super surprising to see that these pathogens were also being influenced by this cascading legacy of cover crop selection. What's become really cool in putting all this together, and again, each of these are independent studies by either my postdocs or graduate students, and I've been trying to take this zoomed out perspective of how it all comes together. What I'm seeing is we're getting to the point where we can start to manipulate these cascades and crosstalk events, where we're getting to a point where we could say, if you plant, sorry, moving stuff around, radish, you can have a depletion of your soil community, which might increase the performance of this type of herbivore, decrease the performance of a predator, increase a below ground herbivore, and have minimal to interesting effects on these aphids. Whereas P could increase your soil community, such as AMF, have no effect on a fall armyworm, nothing on your fusarium pathogens, but might be increasing your corn rootworm and no effect on the aphids. And looking at a triticale, you can have this another effect of augmenting, but reducing the effect of your chewing herbivore pest, increasing the susceptibility to pathogens, no effect on your corn rootworm, and a mixed effect on your aphids. Again, I keep saying this mixed effect on aphids. We did show that data, the volatiles were changing in the plants based on aphid feeding. So we're also going one step further with this to see how predators might be affected by this. And if the volatile odors are changing much in the first example, it means that predators either above or below ground could be affected by this, depending on the cover crop you select. So this is all where we're ending up on our cover crop stories and this manipulating these cascades. We have one more story about defenses to go through and the sequestration of them. So for background on sequestration of plant defenses, it's a green world out there. How come I can't go and chomp on every plant? Well, that's because there's so many defenses that these plants have. One of them are these cardiac glycosides, for example, which we call cardenolides. And a bunch of herbivores, including us, can't really feed or tolerate these things at high level. However, eventually there's these specialist insects that evolve. Not only that can they cope and deal with them, they can sequester them for their own benefit. And you have things that turn into things like the monarch butterfly, where they're now protected from the higher trophic level. And if they're unfortunate enough to end up in the lab of an ecologist, they get fed to blue jays and you have this puking effect. So there's this history of this puking effect and monarchs being co-adapted to these milkweed species. And more, we've been worried about this decline of monarchs overall. And so there's been this slogan about having milkweed and making sure we plant more milkweed to offset the loss that these herbivores are dealing with at various habitat level. But more and more realizing the question is not just having milkweed, it's about having the right milkweed. And this is because, again, plants, particularly plants that have a lot of specialist herbivores, have a bunch of different organisms they have to deal with, whether there's beetles on the roots, on their leaves, aphids, all types of species of caterpillars, and beetles that can affect them in multiple different directions. In addition to that, just in North America alone, there's hundreds of species of milkweed that are across our continent. So not all milkweed are created equal. For example, we can just pick two species here. When we look at things like Asclepias syriaca versus Asclepias tuberosa, that's the common milkweed and butterfly milkweed, we expect to see defenses in these plants. Milkweed gets its name milkweed because when it's damaged, they exude this nasty, sticky white milk, which is latex. If I need to convince you that shooting milk out of your body is a good defense, you can just ask my son for his strategy. Whenever he's carried or perturbed, he will have the same effect. And immediately, the second you touch him and he does this, you want to hand him off and get away from him. So in the same way, it seems like plants have managed to do this, or my son's co-opted this strategy. Fortunately, my son hasn't managed to develop these toxic cardiac glycosides, the cardenolides in plants. So together, latex and cardenolides are a part of this crazy complex or syndrome of defenses milkweeds are expected to have. But as we look across the taxa, we see some plants have high levels of this, such as Syriaca, but think plants like tuberosa have little, if any. So that means not every milkweed plant is equally defended or the same type of, or equally suitable for all butterflies. When we think about this, planting milkweed has been promoted more and more to offset losses in this breeding habitats. So we'll just put more milkweed out there, we'll save all the butterflies. The sad part is we do a lot of this without appreciating all of this host plant defensive chemistry. And I've been in scenarios where I was working in greenhouse or planting milkweeds for different work, and maybe someone might walk by me and be like, oh, that's a great species. I've been putting that in the children's garden. I'm like, no one should be putting this in the children's garden. This destroys the butterflies and is super toxic. But they're like, oh, it has a really pretty flower. And even more so, I started doing searches over my time, and we were looking at these commercial kits that are available, and you can buy these seed mixes that are going to save monarchs. And just even across this, you'll get this 6 seed mix. And through literature and other research I've done across a bunch of different species of milkweed, I was able to calculate there's up to a 36-fold difference in cardenolide content in these seed mix packets. This is important because if you get one of these seed mix packets and you randomly pick a few plants to put out there, these monarchs are going to be growing on these. And depending on this, where they are in the region, up in the northernmost regions, they're going to be feeding on that plant you put out there. And they're going to depend on that plant to build their body, to build their machinery, to help them fly all the way down to Mexico, where they overwinter in these huge broods. When we think about this huge journey that they make, which is over 3,000 kilometers at its longest distance, you have to imagine prepping them up based off the food you give them. In a similar way, we have humans prep up for long journeys when we're running a marathon, right? And we know diet matters. And if you feed on one type of breakfast food versus another, it could potentially affect your performance. I won't say which one's better or worse, but we all have our imagination of how this could affect your performance. Maybe for me, Reese's Pieces is breakfast of the champions. But this isn't totally true analogy when we think about insects, because they're holometabolists. They're kind of hardwired. So the food you give this developing larva is all they're going to utilize to make their adult body. Whereas you or I, we could shift our diet around time, change our muscle mass, and affect our machinery overall. Insects are kind of hardwired and trapped on that. So what they get as a larvae turns into that adult motor, that adult machinery. The better analogy would be imagining if you fed a young child nothing but French fries its entire life, and then you tested how well it performed on a marathon as an adult. So that makes you wonder, what happens when it's time for these adults to fly, depending on their host plant? And we had a few questions in this project that's going on now. And first, we started off with monarch flight performance. And what we were realizing is that there's little data on adult flight, including their metabolic rate overall. We also realized there was no studies on how diet influenced flight performance, and nothing on these phenotypic migrants. So they can have 2 different phenotypes before they go on their journey. We did this across a bunch of different host plants that are common in North America, and then some that have been planted just because enthusiasts like the color of the flowers, and the monarchs seem to grow really well on it. And what we did is we grew all these plants up in the greenhouse, put them in cages with cohorts of larvae. We monitored their development and survival until they pupated. We took the pupae out. We took metrics on their size and weight. We took metrics on the adults that resulted from this, looking at their wing lengths and weighing and sexing them out. We then flew them in these respirometers where we could track their oxygen consumption versus their carbon dioxide output to really get a sense of their metabolism efficiency. And then we could measure things in these by boiling them up and extracting their cardenolides and lipids and looking at their muscle mass and seeing how these things were actually developing based on their host plant. Now, this became a really complicated study with tons of different directions and ways to look at it. But to simplify what we really found in the most simple sense, we found that flight musculature was affected by diet. And we looked at this flight muscle ratio, which is the ratio of their thoracic muscle near their flight compared to their whole body weight. We were seeing that it was affected depending on the plant species they were feeding on. So not all monarchs that were being produced are the same. And it was super dependent on their diet. When we look further at other resources, we took these respiration traces, and this is just an example where we can have them flying in these containers for a while, and we can look at the CO2 concentration over time during their flight bout. What we saw is that overall during active flight, there were significant differences in their flight efficiency or their flight metabolism. We also saw that just even at rest, there's a change in their ability to convert in their consumption of oxygen and output of carbon dioxide. And also we saw significant differences in their cardenolides as well. These were the compounds that they were stealing from the plants. There's a lot more data and slides on this, but for sake of time, I'll wrap it up with just a few more things that we're working on in this. So we're doing this across a bunch of different species. We're looking at all these other things like how their wing beat amplitude are affected. We've tethered them onto flight mills so we can actually look at how long they can fly. That was an old model. I think our newer models look more like this. Let me see if I can click it. And so we have these ability to just track how long they can fly based on their host plant. And we've been adding a bunch of other complex questions to what affects their flight output. One thing we really wanted to do in this part of the study was to go around and collect monarchs in the wild. And we had established these flyaway corridors across the different points where monarchs fly away through and funnel down in Texas before they get to Mexico. And we established collaborators at each state to get all of these monarchs either preserved, where we could measure different markers like troponin T. This is important contractile muscle protein to see how that was affecting musculature efficiency and contracting. We were going to take isotopes to look at their natal origin to figure out where they came from, and do that cardinalite fingerprint again to see what host plant they were on. So we could know how are their muscles, where did they come from, and what plant were they feeding on. Then we could also get living ones, which is really easy to do. I don't know if it's been popular in often North America to release monarchs so you can ship them living. We figured we could get these living ones as well, fly them and do their flight performance, figure out where they came from, and also fingerprint them. COVID put a major dampening on this issue, this attempt, and made it really challenging to do this. But we've also been doing a lot more other studies on top of that. So we've been doing work with a master's student right now in the lab is working at how migratory phenotype might be influenced by host plant and how this could affect their flight performance, how they actually smell and sense and choose plants using GCEAD. And then also looking at this plant defense induction. We're also looking at how neonics interact with larval diet, affecting these three metrics as well. And finally, we were looking at these at the third trophic level and how parasitism through non-target injection of these viruses that parasitoids can have and inject into insects, how that influences this monarchs and these three interacting effects as well with flight performance and plant defenses. So to finally wrap it up with all these crazy stories I told, what am I really studying? So I guess the first question on context and information, I really want to know, how do we use volatile signals and odors? And it depends on how wide a scope you make. You know, if you focus on just one player, you're going to only see that player being affected. But it's important to think of context that we can leverage and manipulate information to our benefit. Another thing with this crosstalk of these asymmetric signals that we studied in talking about this cover crop work was that we know that these signals lead to really interesting asymmetrical effects on herbivores. And they might hint at cool pathways that can either resist some pests while making them susceptible to others, particularly pathogens or other species of insects, depending on their feeding guild or the type of defenses they're susceptible to. Finally, with this sequestration stuff, I'm realizing more and more the combinations of plants and herbivores can either facilitate herbivore performance, whether it's a pest, but also in terms of conservation, like we're doing with the monarch butterfly. And we're ultimately getting at the point that we can see how this could affect the third trophic level as well. All this comes together with fun stories where we use a bunch of cool tools that I love to collaborate with people, where we can sample things above ground and below ground, the way organisms smell, the way they behave. We have a bunch of fun toys where we watch aphids feed or beetles feed on roots, and we can study how organisms move in the soil. We're sampling tons of different plant species for tons of different molecules every day. And although I kind of had some conclusions for my research topic, just to kind of conclude some of those diversity topics, it's important to realize that we're all unique, and it's something that we should appreciate with everyone. But as we try to increase inclusion, not all of us are rare. And from whether it's disabilities, gender, skin color, your native origin, we have to really understand what it means for rarity, for resources, diversity matriculation, and this idea of exceptionalism that can be super harmful as we try to make a more inclusive and diverse environment. Thank you for all sitting through my talk. Here's my lab crew, although we're rarely ever this close anymore. And that's what we look like for the most part. But I hope I have some time for questions if anyone has some.
Well, thank you so much, Dr. Ali, for telling us, so much about your background, and thank you so much for increasing my interest so much in bugs. For our audience, please feel free to type in questions into the Q&A, which is at the bottom of the screen. If for whatever reason you don't want to use the Q&A and you would prefer to say your questions, please use the raise hand button and we'll unmute you so you can say your questions. I'm going to use my position as host and abuse it a little bit and ask my question first. So I was wondering about the volatile signals that plants emit. What's the timeline on that? So say a plant gets pierced and starts emitting salicylic acid. Is it like a seconds thing or a minutes thing or a days thing?
Yeah, we have a bunch of different work on this and volatile responses or overall plant responses. And it really seems to be there's certain molecules that get reduced immediately upon damage. Like mechanical damage, there's a whole suite of volatiles that just get released on tissue disruption because they're sitting there, they have low vapor pressure, and they just get released. Then there's other derived compounds that actually interact with the insect saliva or pathogen cues or these effectors that organisms can have. And they actually cause a breakdown in other metabolites and they release kind of more specific cues. So there's this kind of passive odors that can get released just through damage and they just come out. And there's some that get actually primed by interacting with the actual molecules that the perturber is releasing into the plant. And those tend to evolve with more specificity over time.
That's very cool.
And so it can be from seconds to sometimes hours.
That's very cool. One of the attendees raised their hand. Robin, would you go ahead and unmute them?
Miranda, go ahead. Is it my turn? Can you hear me? Hi, Jared. Fantastic talk. So interesting. I do some similar work with soil feedback, and I'm really interested in the legacy effect you talked about. What do you think's causing the legacy effect? Do you think it's residual volatiles? Do you think it's changes to the microbial community? Have you thought about that?
It's everything. It's like it's almost as though like I've lived, and all of us as academics have lived in a bunch of apartments after it's been vacant. And whoever occupied it first really changes the interaction I have, whether they were tidy, whether they kept the windows open all year long and let residual mildew build up, or if they were really clean. And in a similar way, I think that whole soil community or biome has changed dramatically. And so we initially picked AMF because we could build a hypothesis based off of the plant hormone responses and those crosstalk stories. And they actually turn out to be a red herring. In some cases, the AMF, fit nicely to our kind of expectations, but in others it didn't explain everything. So we've since moved on to look at a lot of microbiome, and we're doing larger and larger soil sequencing to kind of see how that corresponds with all of the responses that we're detecting in these herbivores and pathogens. It's a mixed bag. We take a lot of abiotic measurements for soil quality and soil health. So we have metrics of all of these. We look at nitrogen and phosphorus and all of the full panel of soil key players, and we're seeing some response with nitrogen. It seems to be a complex. Where I'm trying to take away from it is, at least at the first level, is what we can start leveraging for real world recommendations, right? So a farmer doesn't necessarily care whether it was the soil microbiome or if it was jasmine or salicylic acid or which volatile was induced. They want to know that whenever they've been planting radish, their plants are way more susceptible to fall armyworm. And that's not a recommendation cover crops have ever been used for. It's strictly been most of these abiotic type of factors. So we're starting to get to that point, and key to doing that is surveying more and more growers for what cover crops they use and outcomes, and also trying to do more manipulative experiments in different soil contexts across the country to see how different soil types might change the cover crop effect on these herbivores and pathogens and pests. So you can imagine too, if you're doing that soil stuff, there's tons of key players there.
Well, yeah. Can I ask a follow-up? Is that okay?
Yeah, sure.
Yes. Yeah, so I work with a lot of growers, right? And they want me to prescribe a cover crop that'll, protect them against trunk diseases or protect them against cutworms or something like that. So I really liked your the slide you did about protective, right? Like what can you expect to see if you use this cover crop? But what you just said to me in your answer is you're going to expand to look at different soil types. But from perspective, it's so overwhelming because it's soil type, it's climate, it's the microbes that are already there, it's the pathogen you're looking for. Do you think we'll ever be able to have this prescriptive system?
I think it depends on what traits we can detect in the plants too, right? So one thing we're doing on top of this is, so one reliable player we have there is that plant response. And from what we've been phenotyping from the plants, there's ways that we can use those as an indicator for how it's responding to that context. So part of that machine learning stuff with those soil probes and the volatiles we're doing is trying to understand if we can predict the outcomes of either that pest or pathogen based off of the communication, the signals the plant's releasing. So technically we could say like, well, it really looks like this algorithm we're developing, this plant is stressed at this level, and it always leads to an outcome that was negative for a corn rootworm. So we're trying to come up with something that we can kind of quick survey and feed back into a machine learning system that we can kind of make predictions. I think with everything that's complex, it's challenging, but it's definitely something we're trying to do. And if it does resolve and there's more conserved responses, that's something we have to test first. We have no idea how conserved this could be. So if it's highly conserved, the only way to know that is if we test this radish effect across multiple areas and see if we always see this consequence on fall armyworm.
Yeah, that's fascinating. Thank you.
We got a question here in the Q&A from Dr. Mahmood asking which root cell types release volatile terpenes such as terpineol in roots.
That alpha terpineol was from the above ground portion of shoots, actually. So that those studies with the alpha terpineol was in the shoots of plants. We do see a bunch of different terpenoids coming off of the various different root structures. Currently, though, our assay, we don't isolate each root type, right? We take it as the full root phylosphere, I guess, rootosphere. We take everything from the intact root system. So we haven't really pinpointed. What we can do is based on the herbivore, assume where that herbivore was feeding, know that that's likely one of the key places where those volatiles are being released. But systemically, just as above ground, same as below, herbivore feeding on one portion of a root can still change the volatiles of that root system in other portion where it's not feeding. So there's some local things that we could predict based off of where they're feeding, whether on the primary root or fine roots. So we haven't resolved that. I used to do work on that during my PhD, but that was taking me down a different mechanistic dark tunnel. I realized I was like, wait, why do I want to know how I can use this information?
Can I ask a follow-up question?
Yeah.
Thank you. So it's even better if it's in the shoot. I study, by the way, I study plant volatiles. That's what I do at UBCO here. And monoterpenes, including terpineol, usually they are produced in glandular trichomes because normally they are toxic to plant cells. They're very just toxic. They kill the plant cell if there's not. So I'm curious, when induced, is it in the glandular trichomes that expression?
Oh, for the shoots then?
Yeah.
No, we don't.
As well, or is it other cell types that start producing?
We haven't identified. We haven't looked at the cell type directly. We've been doing some work with. Okay, so one part of that is that we are sampling from the complete headspace of the plant. The other thing that we've been starting to tease out are looking at the differences of what happens when you have the stomata closed and open, to see what's getting passively released from those stomatal opening versus the other surface areas. It's very challenging to disentangle the specific portions of plant anatomy. But we do know, again, at least the scale that we have looked at, or there's a history of research on this, is that the systemic signaling differs by local leaf versus systemic or distant leaf. But we haven't like pinpointed specific trichomes or looked at glandular trichomes yet. But there are some papers doing that, particularly in tomato. I've noticed a lot of work coming out of the Rob Last group at Michigan State. And there's a whole trichome group there looking at volatiles and different compounds coming out of tomato plants.
Thank you so much. Excellent talk, by the way. Really enjoyed it. Thank you.
Cool. I see. Is that Karen has a question? I don't know if I can unmute people.
Yes, I can unmute myself because I'm part of the panel here. Thank you so much for joining us today. I was really interested in your comments about exceptionalism and the dangers of assuming it or explaining personal narratives in that direction. So what advice would you have for us how to counteract that and how to better mentor students and friends and colleagues against that particular narrative?
I think what that portion of it, the telling part of that is that I think there's this lack of recognition of the structural and institutional barriers that different groups have, because they're rare and you rarely see them to even hear that story or to recognize what they may be going through. Whether it's the lactating coworker that's struggling to keep their shirt clean and they're getting sick in the bathroom trying to pump milk because there's no resources for them. That's a challenge that's keeping them to want to work at this building anymore or apply for the next grant. Whether it's the person that's in a wheelchair that has challenges getting to the meetings on time because we have an inadequate building. That our systems, particularly in most, like at least from my experience in academia, they select for resistance. And we look across all of the data that show we have a leaky pipeline for all of these marginalized or minoritized groups. One facet is we're missing that point. We just assume if we see a female in the room, all females can do this, right? Not really realizing, and then you just put the onus on people that aren't there and you're just like, oh, they're just not hard workers like so-and-so is. They're just lazy. They're just not trying hard enough, when it's actually huge resource and structural barriers. And we need the systems to stop forcing and selecting resilience. It needs to be more resource and acceptance and understanding that we need to keep the community diverse. It's like, yeah, if I was in a wheelchair and I had to crawl up the steps every day, I probably wouldn't be working at the same level as everyone else until you put an elevator in the building or you make access for me. And those simple ideas of resources change dramatically. You can't assume that person isn't working hard enough if they don't show up and if you don't see them represented in the room. And I think that's something that I've had to deal with as a speaker everywhere I go, because I started to realize this conflating what I might represent. And only until I came across the term, I realized that. Like, I realized a lot of people are like, wow, great, Jared, great to have you here, because now it means that everyone else that's not here, they're just lazy. And it's like, no, it sucks. I saw what happened to my brother. He wasn't late, he was working his ass off. And it's inexcusable that we don't recognize the challenges people are going through. Yeah. So that's kind of what it is. It's recognizing, appreciating, trying to survey and understand what resources are limiting people's performance and why we don't see more equity in the shapes, colors, sizes, types of humans, sex, and orientation that are in the room, and what's holding that back, because there really shouldn't be, that shouldn't be a limiting factor when we just measure our population demographics. Yeah.
Hi, Jared. I have a question about diversity. I'm always really worried about my kids and kids in general being alienated from nature. So it's really interesting to me that you ended up such a hardcore ecologist. Did you have early exposure to nature? Did you have it sounds like you didn't. So I'm just wondering, how did you get pulled into it?
Yeah, I mean, I think that almost by the time you actually look at the output of somebody's CV or what papers they're on, what we all study is pretty obscure, and the pathway seems kind of red. So even if you loved smelling flowers, but then eventually you're looking at carotenoids and petals, it's like, how the heck was I a 3-year-old drawing flowers? Now I'm looking at carotenoids and measuring gene expression. So that windy road is interesting. I think the big key is, like, there's a difference between appreciation for natural history, which most biologists and scientists and humans are missing in general. So that natural appreciation for the appreciation for natural history versus the appreciation for asking questions. And I realized that ecological questions were linking a lot of things that I had a native curiosity about. And so my bigger curiosity was on connections and kind of the sense of there's things we don't see that connect organisms. And I realized that there were actually volatiles that influence the behavior of humans and can dictate and determine who you end up marrying or your likelihood of divorcing that person. or your immunocompatibility, or that plants were releasing things we couldn't see that were affecting higher trophic levels. And so I was kind of mesmerized by this connection between organisms, and that brought me into chemical ecology. To answer my questions, I had to get more and more, I took more and more information from the field to understand the types of connections that were naturally occurring. But I think all of us, if you're a scientist, your passion is for answering questions. And I think you end up going to those places to see where the questions are more interesting. For me, it ended up being in the dirt with nematodes and plants and bugs. But for others, it could be, something like the microbiome of the bottom of our shoe. So I think that it's more about invigorating and empowering people to ask questions in all types of areas. The natural history thing is something that I think takes more time, and we need to cultivate that early on in children, walking in the woods, talking about it. And I think that will play a large role for us caring about our planet moving forward. And I think that's something that is a struggle for all of us. Yeah.
So you didn't have that early love of natural history.
I love bugs for some reason. I like messing with bugs. Like, I think if any of us, I'm surprised not all of us are entomologists. I don't know how we can walk around and not know more about bugs, since they literally smack you in the face. They're likely the first animal you've interacted with, and it's the one that you have more associations with, more fear of, more memories of pain or joy from an insect. But often people imagine like lions and tigers and bears and elephants, where we have these diverse animals everywhere. So there isn't one of us that hasn't had an associated relationship with an insect at one point in our life, whether it was me looking at ants in the kitchen, trailing them outside to the sidewalk and wondering what they were doing, to the weird little times I would make arenas for bugs, and see if it could crawl across a moat and just reroute its way when I was out in the backyard. So I think they were one of the most accessible ones, because they should be super accessible to everyone. For one of us, there's 200 million of them. So it depends on the scale. Monarchs and milkweeds came up way later for my interest, but I was already by then in applied ecology and had learned a lot about natural history to some extent.
Well, it makes sense that a kid who's stuck in the city would gravitate towards nature that they can actually touch and see. So that's awesome. Thank you.
Yeah, and I mean, there's a whole frame of that, and it's cautious to I didn't really live in the city, but it's something that all of us can see, right? So if you did live in a city, you're going to see a bug, and you're going to see trees, you're going to see grass. So you could ask all the same questions, and so anyone can relate to that, whether urban or rural. Have they ever cut a blade of grass and recognized, or cut any plant tissue and realized the volatiles change? So a lot of the questions I ask are super accessible, but I think there's I think the passion for biology and science is about asking questions. And the system didn't seem to be the limiting factor to me.
Thank you so much, Dr. Ali, for your talk and for answering these questions. Thank you to everyone who attended and everyone who asked questions. That's our time for today. So we hope to see you all again next week. And thank you all for attending our very first Diversity in Biology seminar.